Sarah.
Okay, so thank you, Bruno. Thank you, Tim. Thank you all for having me join you. As you know, I am not a member of the report cohorts, but I am here because I believe several meetings ago, you looked around and you said, where are the bacteria? Where are the bugs? So I'm here representing the bugs, or at least I thought I was here representing the bugs until Bruno gave his introduction to the goals of this meeting, which were sort of out of the box brainstorming. And I have to tell you, there is nobody in this world better at drinking caparinos and thinking weird thoughts about TB, especially with young investigators, than me. So if I don't know you, please come with a drink and let's talk. Okay. And I am going to try to keep an eye on time. Who's my timekeeper? Do we have a timekeeper? Oh, it'll show up. Okay, Tim was like very concerned about having time for questions, so. Okay. First, I have a financial disclosure to report. I'm a board member at ONT. I will talk about the whole genome sequencing. None of the whole genome sequencing that I will discuss was done on an ONT platform. Thank you, Illumina. It's all Illumina sequencing, but you should know that. And then, okay, so I, like many of you, have really, my whole career has been motivated by these questions of what drives the diversity of TB infection and treatment outcomes. And I think this is just an amazing time to be thinking about these questions, because what even seemed simple 10 years ago in terms of thinking about the diversity of infection and treatment outcomes, we now understand to be much more complicated and representative of a much greater spectrum of biologic diversity than potentially we had ever anticipated. And that's true both in infection outcomes, but also in treatment outcomes, where we can think about rapid clearers or potentially even post-treatment reinfection as a biologically driven process that would help that we fundamentally don't understand mechanistically. So I'm here repping the bugs, right? And except for the treatment failure in the setting of high-level drug resistance, I think that when we think about these diversity of infection outcomes, the field broadly thinks about that diversity as being the product of the interaction between host and societal factors and environmental factors. And so this is my representation of that. And then there's sort of a little understanding that there might be bacterial diversity that might be contributing to that. But really, the sort of outside of drug resistance, our understanding of bacterial diversity has really been limited to this this sort of model of the lineages of TB, and an understanding that the TB lineages seem to have different epidemiologic manifestations, but we haven't really gotten more mechanistic than that. So from the bacterial point of view, however, what we understand is that mycobacteria are incredibly diverse. They're diverse at a phenotypic level, and they even have genomic diversity, although its genomic diversity is often poo-pooed by people who work on truly variable organisms like HIV. But nonetheless, there is genomic diversity. And there is reason to think that bacterial diversity might be a more important contributor to the diversity of outcomes than we previously appreciated. And so the model we work on in the lab looks more like this. Actually, it's sort of more dimensions than that, but that kind of is as best as my artistic ability could go. So in the lab, we're a genetics and genomics lab. And so we have thought about defining this, or addressing this question using clinical strains. And we started, actually, when I started, I was a straight, like, with Bhavesh, a dyed-in-the-world bacterial geneticist, and it was all about forward bacterial genetics. We're going to do a genetic screen. We're going to define the outcome that we think is important. We'll find the gene, and then, pray to God, we have some way to link it to human TB. So maybe 10 years ago, more than 10 years ago, I flipped my whole lab upside down and I decided we would use clinical diversity as really the motivator for our science, try to understand what how populations of TB were diversified under selective pressure in humans, and therefore using that as our lens into human TB. And we do that using population genomics, sort of standard population genomics approaches, and functional genomics that we then link back to population genomics with the goal of going from clinical manifestations to bacterial processes, bacterial genes, and sort of the molecular mechanisms. And I'm going to give you a high-level view of that today. The goal is not really sort of any mechanistic driver. The goal is to give you a sense of the flavor of that process and some of the ideas that are coming out of this work that I think are really relevant to the report consortium. So our starting point was actually to try to understand bacterial drivers of treatment failure. Clearly, the most important bacterial drivers of treatment failure is high-level drug resistance. We know a fair amount about high-level drug resistance, but we postulated that under 80 years of drug selection, that in addition to the known genetic changes that drive high-level drug resistance, that TB strains have been evolving, acquiring mutations that are causing other drug escape phenotypes. Those might be drug tolerance, those might be sub-MIC resistance, those might be persistence. And by understanding how TB is evolving under drug pressure, we could understand how it is successful under drug pressure and learn how to treat it better. So we've taken a variety of approaches to do this, and we've used GWAS approaches to understand, to identify new genetic determinants of drug tolerance and sub-MIC resistance. But the work that I want to tell you a little bit in detail about is actually work where we discovered a phenotype that we didn't even know existed. So part of the complexity of starting with the clinical populations is when you arrive at a genetic pathway, you know it's important because you know it's evolving in people, but you don't exactly know what it does, right? And so that is sort of the mechanistic puzzle that we try to put together. And so we have used both focus studies of cohorts of TB strains, but we've actually taken advantage of the fact that there are now something over 100,000 TB genomes in the public databases, and you can mine that data to understand which genes are under selection and then try to understand what the selective forces are. So this is work done by Qinyan Liu, whom some of you know, I think. He's fantastic and has his own lab at UNC now. And he took all of the extant TB genomes at the time, yes, little shout out for Qinyan, and used those to identify both fixed variants, that's sort of polymorphisms that distinguish one strain from another strain, and unfixed variants. So those are variants that in a sputum sample, for example, have evolved within a patient to try to understand the long-term and short-term evolutionary forces acting on TB. And we look for evidence of selection by looking for using standard tests. And the one I'm going to show you is actually looking for coding mutations as opposed to non-coding mutations. That's DNDS. And here's what the data look like. So this is on the y-axis. This is the number of independent mutational events. So independently arising, not just one strain acquired at once and then passed it along, but independent mutational events in these 50,000 genomes. The red bar is the number that are coding. The blue bar is non-coding, so that you can see they're heavily selected to be coding variants. And then you would anticipate that drugs have been a very important selective force, so you'd expect to see known drug resistance genes, and all the little black diamonds are the known drug resistance genes. So that was great. And then we also look for intergenic regions because you can't do DNDS, and that's the green box over there. And you can see lots of known drug-resistant genes, but lots of genes under selection without any known relationship to drug resistance, some of which have known relationships to virulence, and we're going to get to those. And so our sort of starting formula is, what do they do? And we just started with the first one on the list, RB1830, which was a transcriptional regulator. Actually, there's a lot of variation in transcriptional regulators that are interesting. And we tried to understand what these clinically emerging variants do. Using a standard formula, we build isogenic allelic variants in the lab using recombinering, and then we just test them for phenotypes. So we tested for drug resistance, no drug resistance. We tested for drug tolerance, no drug tolerance, a little bit of anxiety there. And then Chin-Yun was doing the test for drug tolerance around the Christmas holidays. And when you test for drug tolerance, you exposed to like 100x MIC concentrations of drug, and then you look to see who survives. So you look on your plates to see who survives. And so it was getting very close to Christmas, and Chin-Yun wanted to go on vacation. And so he kept checking his plates obsessively. Now, normally we don't like to do that because normally it's like you're going to get fungal contamination. It's going to be a big deal. But actually, he was up there every day. And what he recognized was these variants actually cause TB strains to recover from drugs faster than wild type strains. So if you expose a TB to 100x concentration of drug, like a fraction of the population dies for 24 hours, a fraction of the population dies. But the cells that don't die, are survivors, and in the absence of drug, they'll start growing again, thus failure. And these bacteria started growing faster. And they started growing faster not only after exposure to isoniazid, but all the first and second line drugs that we tested. And you can see that there. And you could see that both at a colony level and in data that I'm not going to show you at a single cell level using cell wall labeling. And so we called this phenotype antibiotic resilience. And what this was a shortening of the post-antibiotic effect, which I just described for you. It's been recognized in TB and other pathogens for 50 years. Here it is first described in TB in 1969. But mechanistically, it is not understood in TB or any other bacterium. But importantly, our dosing regimens are based on the post-antibiotic effect. And one could imagine a world in which our obsession with simplicity of regimen and making those like giving drugs as few times as possible to make it easier to do DOT, would have selected for variants that are escaping that, and just the selection is around growing faster. And at the time, there weren't that many prospective studies. Thank you, report. This speaks to your importance on that had collected isolates associated with treatment failure, but one of them was the REMOX trial. And in REMOX, pathway variants that created this resilience were more likely to be fixed and emerge and become fixed in this setting of treatment failure and be associated with treatment failure. And it is worth noting, depending on your locale, between 2 and 10% of strains have pathway variants in 3 of the pathways. And I'll just show you these pathways in a minute. So it was this RB1830, which we called ResR, is a transcription factor. And in work that I'm not going to show you, we sort of used classic basic science approaches to map out this whole transcriptional pathway. So ResR binds to 2 areas of the genome that you can see up there. I don't have a pointer, so you just have to RB10C11C and the WEB2 FBIA intergenic regions. They were also under diversifying selection in clinical strains. They had competing binding factors that bound exactly to the same site. Those were also under diversifying selection in clinical strains. The transcription factor WEB2 also under diversifying selection in clinical strains, as is its binding partner WEA. So what we ultimately mapped out was a whole pathway that we didn't know existed that regulates the integration of cell division and chromosomal replication that we think is under drug selection for this pressure to regrow faster after catastrophic stress. Okay, so that's fabulous, but clearly there are other genes on that list that we don't know what they're under selection. And so, oh, sorry, pivot. So that data is looking at TB at a global perspective, right? And actually population genomics approaches, if you have no phenotypes associated with them, require very large numbers of strains. But actually over the past 3 years, we've been collaborating with Report Brazil to use the power of the report cohorts to, and the fact that the strains have been collected prospectively with beautiful metadata, clinical metadata, to obtain power to identify other variants associated with treatment failure. And so in Report Brazil had about 36 isolates associated with bad treatment outcome, only 6 true failure, or 6 pairs of TB isolates associated with true treatment failure, and 30 that were associated with slow clearance. But even within that small number of isolates, actually you have enormous power to find the genetic drivers of failure, because what we did was we took our cue from Dave Lalonde and we actually Cindy measured MICs for all these strains, from the strain that the patient came in with to the strain at 2 months, that if they were slow clearers, and their failure strains. And we also performed contemporaneous whole genome sequencing. And what we found were, in some strains, we found sub-MIC resistance changes associated with new ways of changing known targets. So for example, we found an RPOB promoter variant associated with an increase in MIC to RIF. And we found variants in genes that were currently emerged both in the setting of treatment failure and in slow clearance, representing new putative mechanisms of escaping drug. And here I'm showing you data for variants in the intergenic region between an nitrogen uptake system and a cell wall biosynthetic pathway that Chris Cicetti's lab is now genetically dissecting, and we think alters cell wall metabolism. Okay. So here we go. I just want to see where I am. Okay. Yep. Okay. So treatment and trying to find genetic variants associated with treatment outcome is important, but it's relatively straightforward because those clinical phenotypes are relatively well defined. And in the lab, we understand what the drug phenotype, we understand how to measure bacterial changes in response to drug. But we are really interested in this question of whether there is heterogeneity in the bacterium that is driving some of the diversity in clinical outcomes that we are beginning to appreciate. And that is a more complicated question because we don't even really know how to classify those clinical outcomes. So we were lucky. Oh, but I think there is reason to think that we have underestimated the contribution of the bacterium. And so I want to just remind you that TB is a clonal organism and it's not sexual, right? And so every bacterial strain that is circulating now represents its own successful evolutionary experiment and the history of the selective forces that have acted upon it. And it has not been homogenized through sex with all the other TB strains in the world. And intuitively, we understand that for the lineages, right? We talk about these phylogeographically separated lineages, but this is actually sort of a more profound and higher resolution variation than we typically discuss. And one of the models we've been thinking about in the lab is when we think about these contributions to infection outcome is whether we shouldn't be thinking in terms of sort of this blended model of interacting factors, but whether in fact there has been the evolution of strains that are predisposed to specific subclinical manifestations. So there might be strains that have evolved, their evolutionary success story is cavitation, but there might also be strains, and they're not the same strains, that might, or genetically the same strains, whose evolutionary success story, and for a bacterium that fundamentally means transmission, might be subclinical TB, or might be an altered immune response that we see as what, this resistor phenotype, infected but no interferon producing T cells. And so I want to show you a little bit of data that might speak to this, that comes from a collaboration with Megan Murray and Socios and Salud in Peru. And they had a household contact study where they were collected TB strains from index cases and contacts, right? And I don't have time to go into the whole household contact study, but suffice it to say they did it in 2010 and 2020. And at the same time, they had population level estimates of TB transmission. So what they found was this subclade of lineage 2 strains in Lima, which when they first identified it in the household contact study, they thought was not transmitting because the contacts in the households were not TST converting. But actually, when you did the population genomics, even in the 2010 cohort, you could see population genomic evidence that suggested there was hypertransmissible. And in 2010, it was like 40% of L2 strains in Lima. So by 2020, it was 90+% of L2 strains in Lima, right? So it had swept out all the other L2 strains in Lima, right? Even though in the households, no TST conversions. So for us, that smells like a resistor TB strain. We went on, and we have begun to experimentally test this. If you infect Kramnik mice with this strain, it has a distinct pathologic phenotype, although similar bacterial burdens to its nearest L2 neighbors. But importantly, it has the T cells in those infected mice make less gamma when exposed to APCs infected with either itself or a non-cognate TB strain. So it is a gamma low TB strain. And if you wonder what might be causing that, it only has about 79 genetic SNPs that distinguish it from its nearest neighbor. But importantly, those are in canonical virulence systems. And most strikingly, they're in the ESX1 virulence system. So now for those of you who are not bacteriologists, maybe ESX1 does not speak to you, but ESX1 is the best understood virulence system in TB. It is responsible or has been implicated in basically every pathogenic process that TB is known to execute in survival on macrophages, prevention of phagolysosomal fusion. Its substrates are key targets of the T cell response. And you would not think that the organism would be mucking around with its key virulence system. But what we find in population genomics approaches is that actually there's an enormous amount of ESX1 diversity in lineage 1, the most prevalent lineage in the world, which we do not study. Actually, there are very frequent pre-stop mutations in the core of the ESX1 secretion system. And in fact, 20% of TB strains in the world have ESX1 mutations. We have done some work. Some of those are in the core of it, and some of them are in the key ESX1 regulators. And we have done some work to understand the effect of regulatory variants. And I'm just going to blow through this really quickly so I have time for questions at the end. But suffice it to say, we developed a protocol for very high throughput, cheap RNA-seq of bacterial strains, $5 a sample. And applied that to a set of strains from Lima and from the Oxford Clinical Research Unit in Ho Chi Minh City, Vietnam. So this is 300 strains RNA-seq in duplicate. This is the phylogenetic tree with the gene expression under it. And here I'm showing you expression of the ESX1 locus. And what you can see is there's a lot of phylogenetic diversity in the ESX1 gene expression and recurrent emergence of ESX1 expression variants. And in fact, what we're seeing is recurrent loss of ESX1 or the expression of ESX1. And if you infer transmission on those trees, the strains that are losing ESX1 are actually transmitting more. Okay. And so we wonder why. We have this is like highly speculative. We have our own little like pet model in the lab, which is because it's loss of function, we were able to take advantage of T-and-seq data that generated by Chris Sassetti. And there are 2 mouse strains in which ESX1 variants are more successful. The Inzio mouse and the non-mouse, both models of prediabetes. And so that makes us wonder whether changing human metabolic condition is driving changes in the bacterial population, which is driving its changes in its fundamental virulence strategies. And okay, and here's a challenge for you. So, and we wanted to test this in people, like reality test this hypothesis in people. And the best way to do this right now is this database called TB-Deco. NIH people, thank you very much. This is amazing. But it is actually a database of bacterial genomes with associated clinical metadata. And I don't exactly know the backstory on this, but it's all from Eastern Europe. So there is an enormous, incredibly powerful database, all representing Eastern Europe. And in this, you can see, although you can't read this slide, that there are many ESX1 variants that are associated with diabetes in this database. So in summary, TB is evolving like every organism, even though we tend to think of it as static and homogeneous. The evolutionary solutions may not be exactly what you think, but they have the power to tell us enormous amounts about TB biology. But importantly as well, and this is the power of report, we should not expect those solutions to be universal. We should not expect them to be universal even within or differ just by lineage. There may be highly localized success stories. The Peru strain, we have a Vietnam clade that has a very unique virulent system. And it's the highly local, understanding the highly localized success stories that's going to tell us about the specific and the general biology of TB and how specifically and generally we can intervene. So my proposal to you all is that the strains are low-hanging fruit. Whole genome sequencing is easy. Actually sequence all these strains that you've collected and then develop a shared analysis platform. Actually, Fiocruz has awesome molecular genomics courses and figure out what's evolving in your TB strains across the consortium and then within your local site. And then share that data, because if you don't share that data, what we're going to know is we're going to know a lot about European TB strains. So you got to share that data to make your biology the paradigm in the field. Okay, sorry, that was acknowledging everybody from Report Brazil. Thank you. You guys have questions?
Yes, thank you so much, Dr. Fortune. We'll have questions now. Can someone bring a microphone to I have.
Thanks, Sarah. Super interesting and probably very important. So how are those strains transmitting, do you think? I mean, is it a different mechanism? Are you suggesting, or I guess speculating, that we're moving away from immunopathology in the lungs to the same? I mean, have you got data where you have pathology information?
So I'm speculating without any data that when there are multiple paths to transmission to success. one path to transmission success might be cavitation and pathology and blah, but that in fact, there are strains that have evolved to subclinical TB, to creating the manifestation or tending towards subclinical TB. And that over the population scale, that might be a very effective mechanism of population success. And so, yeah, so that might be that might be an evolutionary solution, a stable evolutionary solution, not just a sort of point on the spectrum towards the more overt signs of TB, and one that we hadn't previously appreciated.
So the business of intra-host variation is really interesting to me. And I wonder if you've had a opportunity to sort of look at regional variations within a single host. In other words, like if I was a resilient strain and I was like suffering under a lesion with bad, unfavorable PK for me, and suddenly I pop into the airway, I might start growing there, or one of the kind of bronchiolitis lesions or something. Do you know anything about whether that variation is sort of just across the whole population in the host, or whether it's different in different spots?
No, okay, it's very difficult to kill people, and
yeah, I guess that's why they have monkeys.
That variation. But the best data that speaks to that is actually from Qinyun and studies drug treatment in China, where you can at least see, and it's hard to know where that is emerging, but you can see the emergence and fixation or collapse of specific variants. And at the time that he did that, it was sort of like random variants, because WEB2, like who knew whether WEB2 was related to anything? But actually what you can see is the emergence and fixation of variants that we now understand are some of these more subtle drug phenotypes. So I think within host evolution is an enormously powerful way to think about what even short-term success looks like for the bacterium, and then short-term success might be increase our power to then understand the long-term success. Yes.
I'm a clinician. I'm not.
I'm a clinician too, a fake doctor.
Yeah. So I was wondering, based in your presentation, that maybe for the reason the bacteria start to grow by the end of therapy, that frightens me. It will be maybe a solution to stop and after restart for a small period. That makes sense for you?
I mean, so I think that is the most compelling model. That if you're taking your drugs every whatever
6 months.
Well, yeah, but it's regrowing a little bit in the pauses, in the gaps, the troughs of your PK/PD curve. So your drug goes up, you're stunned, but then there's a gap, right? And then the bacteria can grow enough to reestablish the population. That's one model. Or the other model is it's sort of at the end, the bacterium has post the 6 months of treatment, the bacterium has the more ability to regrow. Yeah.
Sarah?
Hello.
Super fantastic. It was wonderful. So 2 questions. I mean, this post-antibiotic effect is really interesting. So with the data you have now, can you say more about RESR function in the context of specific clinical backgrounds? So does it interplay with other genetic factors? So that might explain some strain-specific effects we see, with like bedaquiline in lineage 2. Can you say anything with the data you have?
So again, I just speak to report, actually, the importance of thinking about all of your strains, or the power of thinking about all of your strains, is outside of report. There are very few cohorts that give you strain level data and clinical time resolved clinical metadata. And so the idea that you might have strains that intersect with other features, other clinical features, I think is very biologically compelling, but I don't think, we certainly haven't had the data with which to address those questions.
And if I can get another at a more philosophical level, I mean, this idea.
Can I just answer one question though?
Sure.
except cavitation. So I realized, so in work that I didn't show you, we did a PheWAS, phenotype GWAS, and barcoded like 270 of those strains and then phenotyped them in high throughput phenotyping. And out of that, and we used these strains from Vietnam, and out of that, it turned out that there was a little subclade of strains in Vietnam that had very unique experimental phenotypes that were like highly distinct from all the other strains. And if you then went into the Vietnam cohort, those were more likely to be associated with cavitation and treatment failure. Like would make sense because of the link between cavitation and treatment failure. And so that's the kind of link between more complex phenotypes that one would like to make.
But yeah, that brings me to that second question. If you had to speculate, what would be a good composite signature for bacterial heterogeneity, right? So you have genetics. What kind of phenotypic effects should we start capturing when we collect specimens?
I'm a big believer that we don't know what the relevant phenotypes are. And so high dimensional phenotyping, dimensionality is more important than really being certain phenotype X is related to human outcome Y. So to do that study, the phenotyping study that I told you about, we barcoded all these strains and we just grew them in every carbon source we could. Like, do I know what the relevant carbon sources are in people? Not really. We grew them in every carbon source and then we treated with all 9 drugs. And you could see these strains were unique in their carbon preference and unique in their drug responses. You could imagine doing that in even a higher dimension, but that helps you. It's sort of, it appropriates the idea of intermediate trait analysis from human biology. So it says we really think intermediate traits are informative, even if we don't exactly know how they relate. And then ideally, we would find intermediate traits that we could then relate to in vivo biology.
Very cool. Thanks.
Anybody else?
Okay, we have time for one last question before we break.
Hello, Dr. Sarah. I'm JC from Report Philippines. Yeah,
Okay.
it's very interesting because lineage 1 is the predominant strain of MTB in the Philippines and other parts of Southeast Asia. So this is a very wonderful presentation. And we have an ongoing TB pathogenomic study with Vanderbilt University funded by USNIH and the Philippine government. And you gave me a different perspective at looking at ESX1 and RESR mutations, which we previously did not think of. But very wonderful presentation. Thanks to that. Just a technical question. In terms of looking at the mutations at the genomes of these strains, do you do deep sequencing in terms of the whole genome sequencing, and how deep is deep sequencing in that aspect?
So first, can I just give a shout out to Lineage 1? Lineage 1, the biology, the experimental biology of Lineage 1 is different. We don't know, nobody like the proportion of Lineage 1 focused strain or papers is just minuscule to Lineage 2 and 4. And it is the most prevalent lineage in the world. So I just want to like commend that, the emphasis. And it depends on the study. So in all of the analysis of the extant genomes, we just took whatever is there. If we're looking for within host variants, we might go to like 200x. We actually have started looking, using long read sequencing to do try to do more complete genomes, because we understand that SNP variants are not the only like game in town, and there's actually more structural variation than we need to get our heads around. So I think I would say it depends on the question that you're asking.
All right. Yeah. Thank you so much.
Thank you so much.